paper years ago, the University of Wisconsin.
Great to see you.
Well, greetings to everyone. I hope that everyone is doing well, and we're at day 3 of our annual meeting, which is always a good thing. Hopefully you're enjoying things so far. But to get us started, we're going to do another round of our introduction videos. So please enjoy.
Greeting from Senegal. I'm Issa Faye, working with ISRA as a parent trader. Thank you very much.
Hi, I'm Kate Costello. I'm a master's student in David and Soraya Verdioli's lab here at UGA in Athens, Georgia. Right now I'm working on pollinating some peanut plants for crosses to try and introgress some resistance to nice pathogens and pests to make some really great peanuts for the farmers. So.
My name is Nindo Zakara Jaffet. I am a postgraduate student from Kwame Nkrumah University of Science and Technology, and I work under the project Breeding for Leaves for Drought and Oil. Thank you.
Jumbo everyone, this is David Kalolo Kello from Uganda. I'm here in my groundnut research plot, which has been hit by drought for the last 1 month. They are still holding up though. Stay well, stay safe. Peace.
My name is Sean Posey, and I'm a PhD student at the University of Georgia in Agriculture and Applied Economics.
My name is Desmond Sandy Adogoba, a first-year PhD Agriculture Extension student at the University of Ghana. My research area is on the welfare effect of after-led innovation. We're looking at the granule value chain in northern Ghana.
Hello, everyone. My name is Linda Kofi, a first-year PhD student at KNUST studying seed science and technology. My research area is assessing the impact of women-led activities in strengthening the seed value chain of groundnut in Ghana. Thank you.
My name is Ali Mouni Rashim. I'm from Comoros, and I work with Dr. Danielle Fonseca on peanuts.
My name is Precious Tengezo from Malawi. I'm studying Master's of Science in Agronomy at University of Agriculture and Natural Resources. Thank you.
Well, I am Brian Nganda from Malawi, doing Master's program in Agronomy at University of Agriculture and Natural Resources, Bunda campus.
My name is Cecilia Usiri. I'm a Master's student in plant breeding and seed system at Makerere University. Currently, I'm working on assessing seed quality of groundnuts in Uganda. Thank you.
I'm leading the PhD program here at SERAS, and I'm a PI for a different project in the peanut innovation lab. Thank you.
Okay, we hope that you enjoyed those introductions from everyone. And once again, I just want to briefly go over our interpretation for the meeting. We've been offering simultaneous translation in French. And so if you are wanting to enable interpretation, you simply click on the globe and then click on the French channel. So yes, that is all you will be needing to do for today's meeting.
Okay, good. Thanks a lot, Kristen, and good morning, good afternoon, good evening. It's been a really exciting last 2 days, a lot of good presentations and good discussions, and I'm sure today is going to be no different. We've got a very full program today. I just wanted to update you. I know that those of us who were attending yesterday, we ended up the day not being able to complete all the events that we had scheduled. And so we had some discussions after the meeting and agreed with Richard and Justice, who were also to make some presentations in our product life cycle and targeted product profile session, that they agreed that the presentations that we had in that really did a good job of kind of explaining what the concepts are and gave kind of an idea of just how far the different breeding programs in Africa have come. And so we agreed that we didn't really want to reduce the time that we've allocated to our sessions today for our graduate student presentations and particularly for the breaks. So we won't be having those presentations, but we will be making, of course, all of our presentations part of the report and available to all the participants at the end. So we appreciate their understanding and all. And I'm sure there's going to be opportunity in our breakout sessions today for further discussions on target product profiles, et cetera. So looking for good sessions today, and I'm going to turn it over to Jessica, who's going to lead us through the events.
Yeah, thank you, Dave. I'm Jessica Marder-Kenyon. I'm a postdoc researcher at the University of Georgia and the Gender and Youth Advisor for the Peanut Lab. I think a lot of us have had the opportunity to meet each other. We do have a full program, like Dave said, so I won't belabor this too much. We're starting off our program today with presentations by 3 graduate students, master's students that the lab supports. I think as Dave mentioned, on Monday, we support around 64 graduate students and postdocs, primarily in Africa, but also the US through the Peanut Lab. And it's, I think, probably one of the most important and impactful things that we do as a lab. And of course, these graduate students are really the ones that are leading a lot of our research on the ground and responsible for much of the data collection and analysis. And we're very happy to support them as future agricultural researchers and leaders in Africa and elsewhere. We're highlighting the work of 3 of them today. Our first presenter is Jennifer Abogoom, who I think is about to complete her master's in seed science and technology at KNUST in Ghana. So I'll hand it over to Jennifer. I think we've got about 15 minutes for a presentation and then 5 minutes for Q&A.
Yeah, and Jessica, right before Jennifer starts, we are going to quickly do our poll for the day to get everyone thinking and engaged. So I'm about to launch this poll, and it is, what is the biggest cause of poor groundnut yields, which follows along with some of our student presentations. So take about 30 seconds to answer that. It's just always interesting to see some of the choices and what you're thinking. About 30 seconds.
Okay, that's interesting. It looks like lack of proper rainfall, which was also my choice. Slightly beat out the other options, but yeah, clearly there's a spread of different reasons. And I think we all probably would agree that it's a combination of these things depending on the time and place and farm that we're looking at. Yes, as David says, a full answer would be all of them. Thank you, Prof. Okay, Jennifer, are you ready to give your presentation?
Yes.
If you are, you can go ahead and turn your camera on and then mute yourself. And I think Kristen is going to be handling your slides.
Yep, we've got everything showing, so.
Okay, greetings to everyone. My name is Jennifer Abogoom, and I am a master's student in seed science and technology at the Kwame Nkrumah University of Science and Technology in Ghana here. And this presentation is part of my research project, which was to assess the quality of seeds that farmers use in Ghana, as well as to assess their perceptions and preferences about the quality of groundnut seeds that they use in the country. Results of the other activities of the research was presented last year. By then, this study was not ready. And so I'll be presenting on, next please. I'll be presenting on the genetic purity of 2 commercially cultivated peanut varieties in the country. And this work was supervised by Professor Richard Akroma and Dr. David Jordan. In this presentation, I'll take you through a brief background of the study, and then I'll proceed to present on the materials and methods of the study. I will then give the results that was obtained in this study, and then I will conclude and then add some recommendations. Next, please. Over the years, Ghana has experienced an increase in the development and the release of new groundnut varieties in the country. However, studies suggest that less than 5% of the groundnut seeds that are used by farmers in Ghana is certified seeds sourced from the formal sector, which means that the remaining 95% of groundnut seeds that farmers use in Ghana are either from their saved seeds or they get them from the open markets or exchange from other farmers. Now, for the genetic purity of a variety, it's important that you maintain the genetic purity of a variety after it has been released, especially for improved varieties, because it is only when the genetic purity of that variety is maintained that farmers will be able to get or benefit from the improvement that has been bred into that variety. Next, please. Seed quality is an important aspect of crop production for farmers because it helps them in determining the planting value of their seeds even before they go into production. However, and this genetic purity can be determined when you conduct seed quality testing. But in Ghana here, testing the genetic purity of seeds by farmers is rarely done. And this can be most likely attributed to lack of knowledge about how to test their seeds, or they don't have the resources to test their seeds before production. And this was a result that we obtained in our earlier studies in the survey that we conducted with the farmers. So this means that the genetic quality of the peanut varieties that are in the hands of farmers after they have been released are unknown because studies have not really been conducted in determining how the genetic purity of farmers' seeds are currently. Next, please. Chinese is a groundnut variety introduced into a country over 40 years ago, and Yenya also is an improved variety that was released in the country in 2012. Now, from our survey and from reports of other studies, these two varieties are varieties that are cultivated by most of the farmers in the country. And it's because these two varieties are early maturing and they have high market value because Chinese is suitable for soups and all other confectionery products. And Yenya also is resistant to rust, and it also contains 50% oil in the kernels, which makes it suitable for large-scale oil extraction. Now, these two varieties possess the potential to increase the production of peanuts in the country because estimates indicate that about 80 to 90% of the land area which is used for groundnut cultivation is farmers cultivate Chinese in this area of land. And therefore, it's important that it means that these varieties are highly cultivated and highly preferred by farmers in the country. Now, the results of this study is relevant for stakeholders as it will inform them and then about what is happening in relation to the genetic quality of farmer saved seeds, and they will make informed policies and measures that will ensure that farmers plant seeds of high genetic quality. Next, please. So the study was to basically assess the genetic purity of farmer saved seeds of these two varieties. And then in doing that, we sought to use molecular markers to assess the genetic purity of these varieties. because of the advantages that molecular markers offers to assessing genetic purity than the conventional methods. Next, please. As I mentioned earlier, as part of the main research project, we collected 45 seed samples from farmers. And what we did was that anytime we collect seed lots from these farmers, we ask of the names of those varieties. And so we identified 28 of these farmer seed lots to belong to either Chinese or Yenya also. And out of these 28 farmer safe seed lots, 20 were identified by the farmers as Chinese, and 8 of them were identified as Yenya also. And so, for this study, to serve as a reference, we collected seed lots of these 2 varieties from the main Agricultural Research Institute, that's CRI and SARI. We were fortunate to get 2 seed samples from each of the research institute, and then we had 1 Chinese sample from the SARI. Next, please. So for each of the seed lots, we randomly selected 15 seeds, and then we planted them in plastic containers in the plant house for subsequent DNA extraction. And so after 2 weeks of planting, we randomly selected 10 individual plants from each of the seed lots out of the 15. And we took young fresh leaves from these 10 individual plants and isolated genomic DNA from each of them. So at the end of the day, we isolated, our total sample size was 31. And so at the end of the day, we isolated 310 genomic DNA from, isolated genomic DNA from 310 individual plants. Yeah, next, please. So after we extracted the DNA, we checked on the quality and the quantity using a nanospectrometer, and then we diluted it to 100 nanograms per microliter. And so what we did was to pool the DNA of the 10 individual plants representing 1 seed lot. And so we took equal amounts of DNA from the 10 individual plants that represented the 1 seed lot, and then we added them to form a bulk for that particular seed lot. And so the bulk DNA samples was what was then used for the PCR analysis. So at the end of the day, we had 31 bulk 31 bulk DNA samples for our PCR analysis. We obtained 25 SSR primer pairs, which were randomly selected from already existing literature in using it for the PCR analysis. Next, please. So what we did was we initially tested these 25, we screened these 25 primer pairs on the reference seed lots that we obtained. That is the 2 Yenyawosu seed lots from both institutions and the 1 Chinese seed lot from the SARI. What we wanted to do was to identify primers that were able to distinguish between these 2 varieties. And at the end of the day, we had only 4 primers that were able to distinguish between these 2 varieties. And these 4 primers were then subsequently selected and used for the genetic analysis of the 28 farmer-saved seed lots. And so after our PCR was conducted, the amplified products from the about 5 microliters of the PCR reaction were then separated in a 6% polyacrylamide gel. And these gels were run in a vertical electrophoresis unit. Next, please. So all the amplified bands were scored, and each band was considered an allele. And so we scored for the presence and absence of bands manually. And then a similarity matrix was constructed using the simple matching coefficient. And then this similarity coefficient was then used to construct a dendrogram following the unweighted pair group method with arithmetic average. This was done using the Entices software. Next, please. So this is the results of the piece, the gel electrophoresis that we obtained for this particular primer. The first well was the ladder that was used, and the subsequent wells, CH was for the DNA samples that was from the Chinese reference cultivar, and YS represents the Yenyawoso that we obtained from SARI, and YC represented the Yenyawoso cultivar that we obtained from Crop Research Institute. And so from the well labeled 1 to 10 represents to 20, sorry, represents the 20 farmer saved seed lots that belonged to Chayenya, also seed lots that belong. Next, please. So this is a dendrogram that was generated from the cluster analysis. And initially, the cluster analysis grouped all the genotypes into one cluster at a similarity coefficient of 43. But as we move further at a similarity coefficient of 60, these seed samples were then further broken into two main clusters. The cluster 1 basically contains seed lots that belong to the Yenyawoso cultivar, and then the cluster 2 basically contains seed lots that belong to the Chinese cultivars. So in cluster 2, subcluster 2A, we'll find the first CH represents the Chinese cultivar that we obtained from the SARI, and we would see that about 5 of the farmer saved seeds are genetically similar. That is 100% similar to the Chinese cultivar that we obtained as our reference from SARI. And in cluster 1, subcluster A, then we also find Yen S and Yen C, which represents the two Yenyawoso reference cultivars that we collected from Crop Research and SARI. And you would see that about just two of the farmer saved seeds are 100% genetically similar to these two seed lots. Yes, and the remaining seed lots are similar to the reference cultivars at varying similarity indexes. And interestingly, in cluster 2 at subcluster B, you would find up one cultivar which was identified, that is the UWF1, UWF1, which was identified by the farmers to be Yenyawoso, but this variety was clustered under the Chinese cluster. And then in cluster 1, subcluster B, you would also find UEF2, that's the farmers, the cultivar, because these farmers practice have different artificial selection methods in their saving, in the process of saving their seeds for the next season. And so this may affect the quality or the purity of the cultivar during the years of cultivation. And those two seed lots that were clustered in groups other than we expected, maybe because there may have been admixtures in the seed lots, or because these farmers may have identified them wrongly. Next, please. Yeah, as I said, out of the 20 farmer saved Chinese seeds that were used in this study, only 5 of them were 100% genetically similar to the reference Chinese seed lots. And out of the 8 Yenyawoso farmer saved seed lots that were used in this study, just 2 of these were 100% similar to the reference Yenyawoso seed lots. And as I said earlier, 2 of the farmer-saved seeds, UEF2 and UEF1, were grouped in a cluster other than the cluster that we expected them to be found in. Next, please. Next, please. So please go back. Yeah, so in conclusion, our study have revealed that these 2 varieties in the hands of farmers have, the genetic purity of these varieties in the hands of farmers have declined when you compare it to those that we obtain from the research institutes. And so we recommend that smallholder farmers are trained on how to maintain the genetic purity of their seeds after they purchase these seeds from the certified formal sector. And also we recommend that farmers should be sensitized on the need to purchase certified seeds every 2 to 3 years after the first purchase, because most of these farmers, when they purchase the seeds at the point that they are released, they do not go back to repurchase the seeds, and these seeds tend to decline over the years, in genetic purity decline over the years as they cultivate them. Next, please. So I would want to acknowledge the USAID and the BEHEAD program for sponsoring this program. And I would also want to acknowledge my advisors for their support during my study. Thank you very much for your attention.
Thank you so much, Jennifer. That was fascinating and also very on time. We do actually have a few minutes for questions from the audience, and I know that there are a couple in the chat. Jennifer, I'll read them to you because that'll be easier than you searching for them. So Dr. David Bertioli had a question about, well, he first mentioned that he would love to have DNA from the Yenyawoso variety. His question was, is it resistant to late leaf spot and rust? Jennifer, are you there?
Yeah, hello. What I know is that it is resistant to rust, but I will leave it to the groundnut breeders in the country. They would have much information on whether it is resistant to LLS or not. Yeah.
Well,
yeah, go ahead, JS.
It's an interesting talk, but one suggestion to you and your advisor, why you put them together for publication. I really hate you use repeatedly Chinese. I think Chinese, there's a lot of those peanut varieties, like in Shandong province, they have their own, like those code. You should use those code instead of every time it is a Chinese, just like Trump virus, China virus, the same thing. So just be careful for that. When you do science, try to avoid that kind of, no, I don't think it is academic term allowed you use the country as a code for any, like 87% of peanuts in the US are from Georgia, University of Georgia. People will not say Georgia peanuts or US peanuts. You use the code.
Okay. Okay, that's understood, yeah.
Damaris was wondering how many plants of the reference genotypes were used. And she noted that sometimes the contamination can be from the breeder side.
We used equal number of plants, the 10 individual plants from each seed lot, just as in the farmer saved seeds, yeah.
David O'Kello also had a question for you about the gender at sampling, that he noted that women are typically better custodians of seeds than men in his experience. And I'll ask one more question about, also from David, if you noticed any misnomers or other names given to these varieties and the reasons that farmers did that, kind of following up on JS's question.
And please, can you come again?
Yeah, sure. And actually, I mean, David Okello, if you'd like to ask the question yourself and unmute yourself, feel free. You can probably do a better job than I can. I think he was interested in sort of why farmers, if farmers misname these varieties and why.
No, they don't get the seeds. Like I said earlier, most of them don't get the seeds from the formal sector, which would have the formal name for that variety. Some of them obtain them from their colleague farmers or even from the market. And because due to differences in the location, and they give them different names based on the appearance of that variety sometimes. And so this variety may be called differently in one language when you come to one location, but when you move to another location, it becomes a different, then a different name is given to that. So basically most of them may identify, and sometimes it's because the names are difficult, they are too technical for them to mention. They just give them names that is easier for them to mention. Yeah.
Okay, and then Jennifer, just one more question and then we'll take you off the spot. Joseph Gomez was wondering about how farmers perceive the purity of seeds and what methods they use to conserve seed purity.
I don't really have an idea on how they perceive the purity, but I don't think, like I said earlier, in the survey, most of these farmers mentioned that they do not have any knowledge on how to test their seeds. So I'm thinking that equally they do not know the methods or any method that can be used to conserve the purity of the seeds. Yeah.
Great, thank you. That sounds like an important follow-up study. Jennifer, I think we'll move on now to Ivan's presentation, but there are lots of questions and discussion in the chat for you. So you might want to take a look at the chat and feel free to respond there to the questions.
Yeah, and Jessica, I was gonna say one thing. I know a lot of people, sometimes our students, I know Jessica and I see this in our scholarly exchange seminars, they go through a lot to be able to have proper connection and to be able to join us. So while we know that their connection may be in and out sometimes, just be patient and understanding of that because they have done a wonderful job preparing for today's presentation. And if I do see at any time where the audio or connection is going out, we'll be sure to alert them, but we just want to keep them on track and encouraged. And so with that, yeah, Jessica, I'll let you introduce Ivan, and I'll also say Ivan has a little bit of, he's recovering from mild flu, so if his voice goes in and out, he's also working through that, but he's very excited about today. So Jessica?
Yes, so thank you. We have Ivan Chapu, who I think has actually defended his master's in crop science, his research at Makerere University in Uganda, and I think is just awaiting his graduation. So over to you.
Yes, hello everyone. I'm Chapu Ivan. I'm working on high-throughput phenotyping for disease and drought stress selection in groundnuts, but particularly for this presentation, I'll be working on disease. Can you please go to the next slide? Yes, I'm supervised by Dr. Kelo David Kalude, Dr. Kelo Cyrus, and Professor Maria Balota. So particularly for this study, for this presentation, I'll be working on high-throughput phenotyping for breeding in case of groundnuts. Next slide, please. So groundnut is an important crop in Uganda. It's the second most important legume after common beans, currently grown on about 320,000 hectares. However, groundnut production is curtailed by a number of constraints. We have groundnut rosette disease and red leaf spot. These are the most important diseases in Uganda. Groundnut rosette causing about 100% yield loss, and red leaf spot causing about 60% yield loss. Next slide, please. So breeding efforts have been made towards development of resistant and high yielding varieties. And plant breeders always have the problem of phenotyping. Accurate phenotyping is actually needed for to get accurate data for selection in the breeding program. Why is phenotyping a problem? Plant breeders are dealing with very large populations of advanced breeding lines, and phenotyping these populations is usually difficult because traditional phenotyping methods are used. These traditional phenotyping methods involve manual measurement of traits like plant height. You're using traditional rulers to measure the plant height, and you're also using visual scores to assess probably the disease status of the plant. And these methods are subjective, and therefore the data you get is not accurate enough. High throughput phenotyping methods have been suggested for improving the phenotyping process, because with high throughput phenotyping, you get objective measurements, they are faster, and by that you are able to accelerate the breeding process. Next slide. So high throughput phenotyping simply means the use of advanced technologies for fast and accurate data collection by process of automation. So high throughput phenotyping can range from small scale to large scale. On a small scale, you have handheld tools, drones, and satellites. For this particular study, I was working with handheld tools. Next slide. So I was working with handheld tools, 3 high throughput phenotyping devices, the RGB camera, the green seeker, and the thermal camera. So the RGB camera works, these different sensors work with different wavebands of the electromagnetic spectrum. Like the RGB camera works with the visible part of the electromagnetic spectrum. The green seeker works with the near infrared and the visible part of the electromagnetic spectrum. And the thermal camera works with the long wave infrared of the electromagnetic spectrum. Next slide. So these tools have been around for quite a while, and they've been successfully used in breeding programs of wheat, maize, and sorghum, but they have not been tested for use in groundnut breeding programs. So we cannot directly transfer what has worked in wheat and maize directly to groundnuts because of the physiological differences between the crops. Their legumes and the other ones are cereals. So we need to test these tools to see which parameters work best for groundnuts. And besides, we also need to tailor them for the traits we are working with, like groundnut rosette, leaf spot, drought, and yield. Next slide. So the objective of this study is to contribute to the general development and deployment of high throughput phenotyping tools for selection in groundnut breeding. And specifically, my first objective is to assess the phenotypic variation of a population of 50 groundnut genotypes from Equisat, Malawi, and Nasari, Uganda, breeding programs using traditional and HTP methods. 2, to analyze the association between traditional and HTP methods using Pearson correlation and principal component analysis. Next slide. My first hypothesis is that within the population, there is sufficient diversity for biotic stress resistance and agronomic traits to be assessed directly with traditional phenotyping methods or estimated using HTP methods. 2, traditionally assessed plant traits are related with HTP estimated traits. Next slide. So this study is critical for optimization of an application of the HTP tools for routine use in selection during the groundnut breeding, and effectively deployment of these HTP tools for GRD and LRS resistance selection will reduce the cost and time of groundnut variety development. In the long run, HTP tools are cheaper. Resources can actually be allocated to promising lines at harvest. As you know, harvesting is one of the expenses during the breeding process. Next slide. So materials and methods, I was working with 50 genotypes from the Nasari and Equisat breeding programs. So this 50 lanes included all the three available market types: the Valencia, the Spanish, and the Virginia groundnuts. I had two study sites, Nasari, Serere, and Akabango Jinja. All these two study sites, LLS and GRD hotspots in Uganda. So an experiment was planted across the two locations, three replications across 2 seasons. Phenotypic data was collected across 4 different growth points, 5 weeks, 8 weeks, 12 weeks, and 16 weeks after planting. So agronomic traits were collected. We had plant vigor, LLS severity, GRD, pod yield, kernel yield, and biomass. So these were assessed using the traditional phenotyping methods, and HCP traits were collected as well: the canopy temperature, NDVI, and RGB indices. Next slide. So this is how the HTP indices were extracted. So the NDVI was measured using the green seeker. So higher values represent more healthy plants, and lower values represent stressed plants. So, and the flare infrared camera was used for measuring canopy temperature, and the RGB camera was used for taking the RGB photos. Our photo to the right shows one of the photos that has been analyzed using the breed pics of the CIMIT maize scanner. So the green area, that shows the green area with the background all eliminated, only leaving the canopy of the plants. Next. So this slide here shows the different RGB indices we obtained from our RGB image analysis. We have about the maize scanner gives us about 11 different traits, and we also developed 4 different traits, so bring that to a total of 15 different RGB indices. But most specifically, we look at the hue, which is the color of the most dominant color on the image. From 0 degrees, you have red, and 60 degrees, you have yellow, 120 degrees, you have green, and 240 degrees, you have blue. Then we look at the A value, which is a shift from the red to the green in the Scilab color space. Then B, which is a shift from yellow to blue, from blue to yellow in the Scilab color space. Then you have the U and V, which are almost similar to the A and B, only that they're in the Scilab color space. Then we have the green area, which represents green pixels from 60 degrees to 100 degrees hue angle in the Scilab. Then the greener area, which represents pixels from the 80 degrees to 120 degrees in the Scilab color space. And then the crop senescence index, which is basically a difference between the green area and the greener area. So you have a difference between the 60 degrees and 80 degrees, the yellow part, the yellow pixels between the green and the pure green. So the greener area is a more pure part of the green. And next slide. So analysis, we have phenotypic variation. This was done using the ANOVA, using the R package, and Pearson correlation using the R package. Next slide. Next slide, please. So from our analysis of variance, I ran analysis based on the genotypes and different market types. I wanted to see if at all there's variation between the different genotypes and the different market types. So for this study, I'll be presenting mainly the results of the different market types. And from our analysis, we realized that there was no significant difference between the Spanish and the Valencia groundnuts, but the two were significantly different from the Virginia groundnuts for almost all the traits. For plant vigor, the Valencia and the Spanish were more vigorous than the Virginia market types early in the season. For late leaf spot and GRD, we realized the LLS, I mean the Virginia was more resistant than the Spanish and Valencia groundnuts. Next slide. Then for plant height as well, it was the same scenario. The Spanish and Valencia groundnuts were significantly different from the Virginia being shorter than the other two. For plant biomass and pod yield, the Valencia groundnuts, the Virginia groundnuts had higher yields compared to the other two market types. Next slide. So for our HTP parameters, I had analysis across the different time points in the season at 5 weeks, 8 weeks, 12 weeks, and 16 weeks. So for hue and green area, the patterns were similar. Early in the season, the first two data points at 8 weeks and 5 weeks, there was no significant difference among the three market types. But at 12 weeks and 16 weeks, we started seeing significant differences. Virginia groundnuts often had higher hue and green area compared to the Valencia and the Spanish market types for the next two data points from 12 weeks and 6 weeks. Then for crop senescence index, at 8 weeks, we had the lowest crop senescence. That means this is the time where the plants had the most pure green, but all the same, the Virginia groundnuts usually had the lowest crop senescence. That means the Virginia groundnuts were greener compared to the other two market types. Next slide, please. Okay, now the A, when you look at this figure, figure A represents the variation from red to green. So the more positive values represent the red and more negative values represent green. So in this case, we have more negative values towards the end of the season. That means the plants were greener towards the end of the season. Early in the season, the plants were small and there is very little green in the different plots. That's why we had values closer to 0, and that means it was not so green. But towards the end of the season, as usual, Virginia groundnuts are different from the other two market types, and they were more greener compared to the other two market types. So for NDVI, we had different. There were no significant differences for the first two data points at 5 weeks and at 8 weeks. There were no significant differences, but at 12 weeks and at 16 weeks, we begin seeing differences, but still all the same. The Virginia market type had higher values compared to the other two market types. And this is because the Virginia market types have more biomass. All these parameters are accumulated to associated with presence of green biomass within the plot. So the higher the green biomass, the higher the value. So towards the end of the growth season, Virginia groundnuts had more biomass compared to the other two. Then when we look at the canopy temperature, as we got from the flare infrared, for the first two growth data points at 8 weeks and at 12 weeks, there were no significant differences among the three market types. But we have to notice that Virginia groundnuts often had lower canopy temperatures compared to the other two market types. So canopy temperature is associated with the transpirational ability of the crop. So we realize that the Virginia market types were probably able to cool themselves better than the Spanish and the Valencia. Probably this could be attributed probably to the growth architecture of the plant. The Virginia market types often formed a thick canopy and a microenvironment around, and I think this was able to keep enough moisture in the environment around the plants, keeping the canopy temperatures low. And also, it could also be associated with the ability of the plant to extract water from the soil. So we could also say probably the Virginia market types were better than the other two market types in acquiring water from the soil, and were therefore able to transpire throughout the throughout the season, and hence keeping lower canopy temperatures. Next slide. So since we've assessed that there is enough variation for the different traits, for both the agronomic traits and the HTP traits within our population, so we go ahead to carry out a Pearson correlation to see which traits are associated with our agronomic traits of interest. So for this case, we are looking at the pod yield, pod and kernel yield. From our associations, I realized the best associations we get really was best related to parameters collected at 8 weeks. So the greener, we see green area and greener area having the highest correlation with pod yield. And this simply means that plants which had higher, more green biomass within their plots had higher yield at the end of the season. This is logical because green area is associated with the photosynthetic ability of the canopy. So a plant which had higher green area probably was able to fix enough food to support enough pods at the end of the season. Then we also look at the canopy temperature. Plants which had lower canopy temperature often had higher yield at the end of the season. Next slide. For LLS, we look at NDVI. NDVI was the most sensitive and it had the highest correlation with LLS severity. So the correlation was negative. That means increase in the severity decreased the NDVI values, because as the severity increases, there's defoliation of the leaves, and loss of the leaves reduces the reflectance ability of the canopy. Next slide. So then we look at the groundnut rosette disease, GRD. For GRD, we realized that NDVI still was very sensitive compared to other parameters. Then also the green area and greener area were very sensitive. So groundnut rosette is associated with stunting of the plant, and by stunting and rosetting of the plant, the canopy size reduces. And once the canopy size reduces, the green area covered by the plant severely decreases, and this can be assessed using the green area. So plants with healthier plants had bigger canopies compared to the affected plants. Next slide. And so for biomass, there was a positive correlation between NDVI and green area with the biomass. So plants which had more biomass had higher values for NDVI and larger green areas. And there was a negative correlation between the A value, which is the greenness of the canopy, and the greenness of the canopy, and the biomass. So greener canopies, plants which were deeply green often had higher biomass. Okay, next slide. Okay, let me conclude. So from this study, I realized that there is adequate variation for both agronomic and HTP traits within the groundnut population, and and the different market types differed significantly, and HTP parameters were highly associated with visual scores used by the breeders. So the HTP measurements can be used to complement the traditional measurements used by the breeders, or if at best we get better predictions, we can even be able to completely replace them in the future. And yield prediction is actually possible. As early as 8 weeks, a breeder can be able to tell which genotypes are going to perform better at the end of the season. Yes, next slide, please. So there is still work going. There is still ongoing work. I'll have to assess the heritability of these HTP parameters, which are highly correlated with the agronomic parameters, And I will have to develop models and validate the regression models for active deployment for selection in the breeding programs. Next slide. Yeah, I would like to thank my supervisors, Dr. Maria Kalule, Dr. Maria Balota, David Okelo Kalule, and Cyrus for the help, and the Innovation Lab for all the assistance. And particularly, I think I've never thanked Jessica enough. She's the one who delivered these machines to me in Uganda. Thank you very much. Yeah.
Thanks, Ivan. That was fantastic. Congratulations on this work. I think you'll see in the chat you have a very impressed audience. So yeah, we're very proud of you. Just to keep us on time, I think that we will move into Danielle's presentation now, but Ivan, there are some questions in the chat box. And so I would encourage you to take a look at those and feel free to continue answering questions over there. So our third and final student today is Danielle Essandoh. She's also successfully completed her master's degree program in plant breeding and seed systems, also at Makerere University in Uganda, and is currently awaiting her graduation. So Danielle, over to you.
Hello, everyone. My name is Danielle Essandoh, and I'm presenting, there's a little change in the topic, but it's the performance of groundnut interspecific lines of Arachi species in Uganda, and identification of quantitative trait loci associated with disease resistances. I was supervised by Dr. Soraya Betioli, Dr. Thomas Odong, and Dr. Okello David. Next slide. Okay, so groundnut is a very important food crop in Uganda. It's the second most important leguminous crop after common beans, with an average coverage of 430,000 hectares. The major growing regions are in the eastern and northern part of Uganda, and serves as a major crop of economic importance. Next slide. Okay, so despite the importance of groundnut, the crop is affected by several constraints, amongst which are abiotic and biotic factors. Amongst the biotic factors, mainly in Uganda, is late leaf spots, which causes an average yield loss of 60% annually, and groundnut rosette disease is causing average yield losses of 100% disease annually. So for A and B shows the defoliation of leaves by late leaf spot, and C and D actually gives us an idea of some of the symptoms of groundnut rosette disease. Next slide. So over the years, the groundnut breeding program in Uganda has used conventional breeding approaches to improve resistances. We've actually, Dr. Kalule has actually released several good lines, such as the Serenut series and the Narrownut. However, some of the lines are still succumbing to disease pressure. An example is Serenut 4, which was initially resistant to GRD disease, but then succumbed to GRD under high disease pressure. Also, the origin and isolation of the groundnut crop from its diploid parents have further narrowed the genetic base of the crop, therefore reducing allelic combination for resistance. It also restricts breeding. So this is actually how the cultivated groundnut was obtained, the figure on the left. Next slide. So wild species have been used in several breeding programs to widen the genetic base of crops, including chickpea, soybean, common bean, and even groundnut. So the only problem with using wild species over the years has been the differences in ploidy levels, the differences in ploidy. However, interspecific lines, which consist of advanced backcross QTL lines and chromosome segment substitution lines, were obtained from collaborators in Senegal, specifically Syrah, through backcrossing with a recurrent parent FLE11. These lines introduce wild alleles from 4 different wild species and could actually be used as parents in crosses. From the previous presenter, Ivan, we realized that phenotyping is also mostly does not give you precise results and time-consuming and expensive. Therefore, we need to also identify QTLs associated with disease resistance, which will save time and also expenses. Next slide. Okay, so despite the fact that several interspecific lines are available and can be tapped and used into the breeding program in Uganda, little progress has been made in incorporating them in breeding programs. This is actually because one, there's limited information about the agronomic potential and responses to groundnut rosette disease and late leaf spots, which are major diseases in Uganda, and it will also inform their usefulness as parent. QTL identification is one of the prerequisites for marker-assisted selection, and it's unknown whether there are QTLs associated with groundnut rosette disease and late leaf spots amongst the interspecific lines. Next slide. So my general objective is to contribute to increasing the potential of cultivated groundnut varieties in Uganda using wild-derived lines. Specifically, I seek to determine the response of interspecific lines to groundnut rosette disease and late leaf spot, and also to identify quantitative trait loci associated with resistance to late leaf spot and groundnut rosette disease amongst the interspecific lines. Next slide. So I hypothesize that the lines outperform our local checks for resistance to late leaf spot and groundnut rosette disease. And secondly, QTLs are associated with the late leaf spot and groundnut rosette resistance due to the wild introgressions. Next slide. Okay, so when we identify interspecific lines with diverse agronomic traits and resistances, these actually will serve as novel alleles for stresses and agronomic attributes, and there's also a bridge between our wild and cultivated groundnuts, therefore making it easy to make crosses. When we also identify QTLs associated with resistances, will actually guide decisions in developing molecular assays and also further, in the long run, reduce phenotyping cost. Next slide. So for the first study, which was to evaluate the lines for responses to diseases, 3 populations were used. And the first, each of them varied in the wild combination. The first was from A. valida, second Patizoko, and third Ipiensis, of which the numbers are given. So DOC-IR and DOC-IT were resistant checks for groundnut rosette disease and late leaf spots, and they've also been reported to have wild introgressions from A. digoe. Serena-2 is a resistant check for groundnut rosette disease and late leaf spots, and also the check for yield in Uganda. FLAIR 11 is the recurrent parent from which the interspecific lines were generated from. Next slide. So in all, the lines were 376, with the, excluding the checks, and were evaluated in 2 hotspots of groundnut rosette and late leaf spots using a 19 by 20 alpha lattice, 2 replications and 2 seasons. Data was collected at 4, 8, 12 weeks and at harvest, and also some yield parameters were taken. Percentage disease incidence, which was taken for groundnut rosette disease, which was estimated as number of plants infected divided by plant stand count by 100. The data was then fitted to mixed linear models using the LME function in R. And the severity scales are actually given on the right tables. The late leaf spot severity scale was used as given by Subramaya et al. 1995, and severity scale was based on Wali et al. 2007. Next slide. Okay, so this is just the summary of the analysis of variance for disease and yield parameters. This is just to show that the genotype mean square was significant were significant for all the traits, indication that there's some amount of variability amongst the lines for late leaf spot and groundnut rosette disease and yield, warranting easy selection. This is actually supported by the heritability estimates that were moderate for all the traits, an indication that the lines could actually be selected based on phenotypic variation. Also, the heritability estimates also kind of give an idea of the at what stage should optimum evaluation be done, and you realize that optimum evaluation could actually be done for JRD at severity and late leaf spot at harvest. Next slide. Okay, so this graph actually is just to compare how our interspecific lines did in comparison with our local checks. So the x-axis gives the JRD severity, and the y-axis gives the pod yield. So why are we comparing groundnut rosette and pod yield? This is actually because a strong negative correlation was observed between groundnut rosette disease and pod yield in comparison with late leaf spots, indication that the lower yields we were viewing or seeing was actually due to groundnut rosette disease. So you find that our lines were the checks, the Serenat 2, the dog IT, and dog IR, did relatively well for groundnut rosette disease. They were highly resistant in comparison with our local checks. But if you look at where the blue circle is, you realize that several of the lines actually, about 8 of them performed our best local check for yield, indication that there's still room for improvement of some of our local lines, and we can actually tap some of these alleles from the interspecific lines into our local varieties. However, it would actually be very helpful to elucidate the mechanism of resistance that are acting in these lines, including the checks, as GRAV, the helping virus, has been reported to cause yield losses, despite the fact that it does not show any symptoms. Next slide. So this is just the comparison of our interspecific lines with local checks for yield. So for the first graph to our left, you find that several of the lines had lower days to 50% flower, which gives an indication, is also a measure or indication of early maturity. And you find that they did outperform our local Serenat 2, an indication that we could actually start initiate a crossing program to integrate some of these good yield traits into the background of some of our local lines, despite the fact that they might not be that resistant to groundnut rosette disease and late leaf spot. And next slide. Okay, so identification for the second objective, which was identification of quantitative trait loci associated with resistance to late leaf spot and groundnut rosette disease. 133 interspecific lines from the Abatizokoi population was used, and the phenotypic data obtained from this study, from study 1, which was actually my second objective for my thesis, was used. So all statistical analysis were performed using the R statistical programming language. And the physical map in conjunction with the phenotypic data was used for QTL identification using the RQTL software. And the percentage of phenotypic variability explained by the QTL and the estimated effect was assessed using the fit QTL function in RQTL. Next slide. So this is the high dense linkage map that was obtained and used for my study. This was actually obtained for the Abatizokoi population, consisting of 140 SNP markers. Next slide. Okay, so for late leaf spot, this table actually gives us the QTLs for late leaf spot in the Abatizokoi population. So for late leaf spot, two QTLs were identified, which had relatively lower phenotypic contributions of less than 10. However, it's been in literature, it's actually been reported that the genetics of resistance to late leaf spot is complex and controlled by several major QTLs. So the QTLs obtained in this study were minor and could actually be validated and used in the breeding program. Next slide, please. Okay, so for groundnut rosette disease, similarly, 4 QTLs were identified. These QTLs are novel and have never been reported in literature, and 3 of them who had moderate phenotypic variation explained and are good candidates for selecting for groundnut rosette disease in Uganda and elsewhere and should therefore be validated. Next slide. Okay, so the QTL effect plot for late leaf spot and groundnut rosette disease was actually done to determine the contribution of the wild alleles in the background of the FLAIR 11. So generally, almost all of them had lower trait values for disease resistance being contributed by the wild alleles, which actually confirms the value of wild species as sources of resistance in the breeding program, with the exception of GRD severity, which had higher trait values for disease resistance due to the wild alleles, which also actually confirms that some wild alleles come with negative traits. Next slide. So in conclusion, 17 resistant lines were recorded from the A validator population. and 6 lines, 2 of which were from the A. batizokoi population and A. valida population were resistant to late leaf spots, whereas 70 were resistant to groundnut rosette disease. Amongst these, these two lines, the lines, actually combine high yields into a disease background than the local checks. The QTLs identified for late leaf spots and GRD disease also provides additional tools for marker-assisted selection and therefore require further validation. Recommendation. There was so much variability amongst the line. A study was taken up because of the limitation of time, but lines were very variable, especially for various yield parameters. 12 yield parameters were taken prior to the study and they were very variable. Also, a coursing program has already been initiated at Nasari to incorporate some of these cryptic alleles into the background of some of these lines. And I also recommend that a coursing program should be done to make crosses between our lines and some of these wild synthetic allotetraploids, specifically for disease resistance. Next slide. So I'd want to acknowledge the following organizations and specifically my supervisors, Dr. Soraya Betioli, Dr. Odong, Dr. Kale, Dr. David Betioli, Dr. Carolina Balen, and Dr. Carolina Shavaro. Thank you so much.
Thank you so much, Danielle. That was great. There is a question for you in the chat from Sham. Can you see it? I'll let you take a look at it and respond. If you don't see it, let me know and I'll read it out to you.
Okay, I can't see it.
Jessica, can you repeat, can you ask Danielle the question?
Yeah, sure. So the question is, do you know the actual accessions of the different wild species used in this study? We know that not all accessions of a wild species are resistant to a particular disease and exhibit significant variation. So it's important to list the accession/collector number of the species rather than just using the scientific name A. valida or A. geranensis.
Okay.
And then I think Isa was also pointing out that it seemed to him that GRD might not have been as severe in your environment because most of the lines were scored below 3.
Okay. Jessica, should I answer the question?
Yeah, please do. Yes.
Okay, so for Isafe, yes, the GRD severity was severe. I think for some of the seasons they were not, and it's actually been reported in literature. It's been reported as so for instance, 2019B in Uganda, you find low GRD severity, and 2020, the A season usually
Jessica, it seems that Danielle's audio went out. So Danielle, if you are able to answer a question, feel free to do so in chat, but we'll also be sure to email you any of these questions.
Yeah, that sounds good. I'll just say a couple things before we head to our break. Just, so that everyone's aware, I think, of how impressive this work is, that they also managed to complete these masters and do all of this research in the middle of a pandemic during pretty severe lockdowns at different points in their program. So we really do applaud these 3 students and all the rest of them for having managed to do this work so successfully. I also wanted to note that we, like I said, these are 3 out of 64 of our graduate students and postdocs in the lab. And I think that we are planning to host an event later this summer where we'll have all of our graduate students present. So we're looking forward to that. And of course, we would be happy if you all joined us for that. And then also just a reminder that we, this year in 2021, at the beginning of the year, organized a monthly scholarly exchange seminar that sort of focuses on different aspects of professionalization for young scientists and researchers. I know Helga and Cynthia and a couple of other, Farid also from our EAP, have joined us at those meetings, and you know, we're continuing to kind of develop that programming, and if any of you would like to participate in that, we would appreciate it. I think just before we get to the break, Allison was hoping that we would do a group picture.
Yes.
So, well,
I
yes.
would love if everyone would be able to turn on their camera for just a moment. I'll watch people pop up, pop up, pop up. And then I'm going to take a couple of shots before people start wandering away to take a break and eat lunch and run to the restroom and things like that. Okay, we've got just a second. Anyone else? We've got about 3/4 of people. Wow. Okay. All right, I'm gonna start taking some shots. Everybody look your most photogenic. You ready? A few more pictures. Pictures, pictures, don't move. It's great to see everybody's faces for just a minute, isn't it? Just a moment. One more. Then I promise I'll be done. Okay, I'm done.
Great, thank you, Allison. I'm looking forward to seeing those later. Yeah, so we're running pretty much right on time, even a little bit early. So I think we'll take a break, show some videos, and then I think we're reconvening at
around 11:30. Yeah.
Yeah, on the 30, wherever you are. So we'll see you back here in a little bit. Thanks. Okay, welcome back from the break. I hope that everyone was able to relax or get something to drink in the meantime. We're moving on to our last section of our annual research meeting, which are these breakout discussions.
Yes.
I'll let Kristen walk us through the logistics.
Yes, we do have quite a few logistics to cover for our breakout rooms. So, as y'all know, this has been a learning process for our virtual meetings, so hopefully they will go well. I will say to my moderators and rapporteurs, you have already been assigned to your breakout rooms. For our other attendees, you are going to have the option to pick which breakout room you would like to attend today. So we do have quite a few rooms. I'm going to tell you their names. We have bottlenecks and groundnut seed systems, applications of remote sensing and groundnut, priorities for groundnut nutrition research, forging partnerships between USAID and country programs and innovation labs. We have rethinking research priorities for youth in agriculture. We also have adding gender responsiveness to research successes and failures, as well as technology pipelines and PLCs for social science projects. So,
welcome back, everybody. I think we are slowly making our way back into the main room.
I got cut off.
yeah, we probably could have taken even longer to have more discussion. So
definitely, I'm not sure we even got through our first question.
It's okay.
I think we've got about 30 minutes now to kind of report back.
Yeah, I mean,
from each of our breakout rooms, yeah, and maybe continue the discussion.
You have a little bit. I mean, we obviously, yeah. I mean, we hopefully we'll try to finish by 1. I don't have any real major significant closing remarks, but I think, yeah, I'll let you sum up everything.
Yeah. Okay. Well, I guess we can just go down the list. Yeah. I mean, just our first. That's you, Jamie. Our first breakout room was about bottlenecks in groundnut seed systems. I think Jamie was the moderator. I'm not sure who your rapporteur was for that room, but if you guys are ready to share in a few minutes what you discussed.
Yes, it's Jamie and David Okello.
Yeah,
So Jamie, our moderator, and David Okello was our rapporteur.
We had a nice freewheeling conversation over a very big topic. So I think we had some good thoughts in there, and I'll throw David under the bus to summarize.
Yeah. Okay. Thanks. Thank you for entrusting me in all this. So it is all about me now. Whether I got the right one or not. The first topic we discussed was what are the bottlenecks of the groundnut seed systems. I must say we could have gone over and over again. There are lots of points still keep coming up, and I still would still wish that they could follow up with this team to continue bringing out some of the issues. But these are some of the key points which came out, that the seed impurities from the seed companies moving down to farmers. This one of the bottlenecks, and then this one affects the kind of product and the quality which the processors come up with. Then of course, with the poor seed quality coming from the seed sellers, there is low incentive by those. That's why some of the farmers resort to land races. And then the longer time of seed saving, probably the producers tend to keep it longer than expected, and the maintaining of the purity in storage. Probably we realize that some of the impurities come from the storage conditions, and then also at the shellers. These are also areas where impurities would come in, and then the cross-contamination of some of the classes, of seed. For example, high lake. This one is going to become a big problem. Lack of actual demand to guide the supply, the issue of pests and diseases, and unfavorable weather, and then low germination rates. And then, of course, no clear-cut differences between seed and grain producers. Those were some of the bottlenecks we raised in there. And then some of the key challenges or innovation which could inform the early generation seed production are, if we could collect the actual demand to guide the supply, that could be a good, baseline. Then if we could register the seed growers and the who are frequently monitored about quality.
And then incentive to quality, probably if, paying premium would be given, that could probably make people have several things. Then probably some measures, punitive measures to, seed quality. In Uganda, for example, here, we had been proposing that some of these companies could be deregistered if they, they're caught doing that. And then we need to promote both formal and informal seed sector. We realize that actually dual system exists, and in Africa, for instance, the informal seed sector actually is contribute over 80%. So yet the more investment come from the formal side. And then we could also introduce some classes of seeds, like quality declared seed is common now in Asia and also in Africa. And then also we could accept some cycles of seed, for example, foundation seed cycle 1, 2, certified seed cycle 1, 2. This would help actually beef up the volume since groundnut is having low multiplication ratio. Then the third question was what could be done, what research could be done to help improve the seed systems. So probably one of them was that frequent early generation seed multiplication to further multiplication pathways. We realize that early generation seed is the owners of the institution or the producers of the seed. So if they can beef up their multiplication possibly down in the pathways, the seed volume would increase. And then a little bit of mechanization of the farm operations, for example, shelling, sorting, branding. This could go a long way, improving the early generation seed. And then a strict certification regime, investing into a financial credit and insurance, probably that would give incentive to the producers. And then also investing into ICT, for example, mapping out farmers using some app and having dashboard where, it's called like a market space. We have seen this in Uganda, for example, in ISSD. They have something like that, which farmers are mapped. They know their monitoring regime. And this could go a long way, for example, improving the EGS. Then investment in storage. Most of the breeding programs don't have cold storage. And then, of course, also just storing of the normal seed for short to medium term. So those could be the investment areas or researchable areas. These are some of the points I managed to capture. Could be, if time allows, some members feel that some of the stuff are left out. This is the time for them to come up. Thanks.
Thanks, David. Yeah, we do actually. If other people would like to add, we certainly have a couple minutes to do that.
David, are you going to send these to the management entity so that we can get a hold of them?
They are indeed.
They are indeed?
Mm-hmm.
Good.
Yeah. I'm going to post all this to them.
That was quite a list, David. I don't know that I have anything. If there's any others in our group, it was, it's hard to separate in some ways what we were just talking about versus what especially breeders who are engaged with this deal with every day and think about. So I know there's a lot of thought there, and we do need to think about some innovative ways to move forward on that, because we are not getting our varieties out there. And then I'm really worried about getting some of them out there and then losing those technologies as well. We're hearing stories about, oh, we replaced this one variety, but then the farmers see it and they say, oh, it's not as resistant, but it could just be seed purity issues. And at the same time, we break trust with the farmers to do that. So it's a compound issue for sure.
Great, thank you guys. Yeah, we will definitely gather all these notes together and then share them with everybody. Austin, would you like to run us through what your group, or sorry, whoever your rapporteur is, I think maybe Peggy, who's unmuted, to talk about their discussion on applications of remote sensing in groundnut.
Yeah, Austin, I don't know if you want to say anything or you want me to just jump in.
Go for it, jump in. I'll add on to the end if there's anything.
Okay.
Great.
So this is not my field, so feel free to add on. So our assigned topic was remote sensing in groundnut, but we really made the discussion quite a bit broader, and it was more about how digital agriculture can actually be an asset to farmers. So it's much broader than remote sensing. It's even much broader than just production data. If we're really moving eventually towards digital agriculture, it also could include economic data, market data, et cetera. So one of the questions is about whether farmers will actually use the data. So one of the objectives of the remote sensing, of course, is to be able to predict primarily yield, but then in particular in Africa and, well, anywhere where it's a problem, aflatoxin levels or other disease incidences using remote sensing data, or perhaps even early in the season, just crop stand and replanting. So there were two issues that were discussed. One is how actually to communicate those data to the farmers. And then the other question is to the extent that they might use the data. And so communicating the data to them, a point was made that in some sort of visual fashion, using maps or images makes it much clearer for them to actually adopt the information or use the information to make decisions. So ultimately, it's about some sort of decision support. And whether that can be built into apps, and it certainly has been in a lot of situations, but maybe not those that are particularly applicable to some of the agricultural scenarios in Africa. In addition, it's seen that farmers everywhere may be skeptical of some of the information. And so how to actually get them to trust the information or to see the benefit of the technology. In some cases, there is, of course, a question about the cost benefit. And so will it be worth it to actually use that information? Now, since much of this is just beginning in the Peanut Innovation Lab projects, the participants in the session thought that it was very important to engage with the local communities. So basically to include them in participatory research, actually have farmers collect data sets, perhaps collect images that could be submitted, and then actually communicate then how the data were being used, what the outcomes might be, and then how might they use that information to inform decisions on perhaps earlier harvest if it's an issue with aflatoxin contamination and it's going to be predicted to be a bad year, would they want to go in and harvest early? Of course, it was realized that there are many other issues associated with simply getting the crop out of the field. So there are gender issues, labor issues. Peanut is not a monoculture in a lot of cases, or there may be other crops that are also being cultivated by the farmers that may take precedence. There are markets to consider. So it's a very complex issue. And so it needs an integration of all of these factors rather than an isolation of just using the remote sensing data to actually make a decision. I think that was pretty much what I captured, but feel free to add to that.
Awesome. Thanks, Peggy. And it was a broad topic, and there is a lot of moving parts going into this. And trust is such a big factor in the adoption of these techniques. But one of the things is the work that Ivan's doing with the high throughput phenotyping, work like that, and working with those, being able to work with individual farmers in the PIL's efforts, I think ultimately will help lay some groundwork for landscape, regional scale analysis. So the work that's being done in the fields can ultimately be really great work for being able to monitor these groundnut fields across Africa. So I think there's a lot of promising future to this field. And it seems like every year there's a new remote sensing platform available. So, and we're only getting more and more accurate, higher detail information. So I definitely think that this remote sensing realm is just really scratching the surface. So huge thanks to everyone who participated in our session, though. It was a really good conversation, and we hope we can continue it in the coming years.
Yeah, I think a lot of us have had, certainly from our group as well, if we want to keep this conversation going. And so whether that's through, organizing other kind of standalone discussions like this, but yeah, this is all important stuff. Okay, yeah, let's move on to Mark Manere's group, which was looking at priorities for groundnut nutrition research. I'm not quite sure who the rapporteur was for that, but if Mark or the rapporteur would like to go ahead, feel free.
Yes, Isa was the rapporteur. So.
Great. Isa, do you want to share with the group a few things that we talked about?
Yes, you can go. Yes, please. Because I have captured some. Yeah, go ahead.
Sure. I mean, I think we touched on a couple of issues. None of them were terrible, were entirely novel, but the fact that they're still around means that we still have work to do about them. And that is one is the reality and the I guess I would use the word myth about aflatoxin. So we still are burdened by a strong opinion when we don't know if that opinion is just in the public, but it also may exist in lots of realms in USAID about aflatoxin being dangerous and how much aflatoxin is dangerous, and peanuts. The way you can just avoid aflatoxin is avoiding peanuts, things like that. So, yeah, just raising that as an issue of concern. What there seem to be plenty of research projects in some level, maybe not enough, but maybe the barrier, the activation energy that we're having trouble overcoming is how does that translate into peanuts going into people's bodies to generate better health and nutrition. So, whether that is through secondary agencies, like the, in the focus of PIL, of course, is in countries that are subject to, focus of USAID interventions. So I think that is either through other USAID sponsored projects or from the local governments as well. And maybe getting a sense of what kind of research would be needed for more, what do you want to call it? Widespread usage of peanuts, I guess. So that's pretty much the things we talked about.
Great, thank you, Mark. Isa, did you have anything else to add, or anyone else from that group?
Also to add about what we talked about the need to assess or to screen the germplasm we have collected for quality trait related to nutrition, because we know that in Africa we have some varieties that are being used for different utilization, and if we want to develop the peanut industry, we need to assess their quality trait profile. We don't have enough information regarding the quality traits like the oleic content. We don't have also information regarding the vitamins, regarding also the protein contents, and so. This information will be very important for breeding programs. And we also discussed the fact that we know the relation that may have between the aflatoxin contamination and the drought. But most of the breeding programs in Africa don't have the equipment or the facility to take into account the aflatoxin as a criteria or a criterion in their breeding program. So there is a need to improve the skills and also the equipment of the breeding programs to make sure that at least we're gonna have more tolerant or more resistant varieties, because we know that aflatoxin contamination is the most is a very serious challenge that we have in Africa because of the warm conditions that are very favorable to the development of the fungi. I think if other colleagues have something to add, they can do. Thank you.
Yeah, thanks very much, Issa. Those are really important points, I think. Does anybody else from the group have anything to say?
Well, I add some little things for that. I think actually raised a good issue about that. When we talk about the peanut nutrition, we want to, the final goal for this program is to improve the quality and the quantity in the people eating the peanuts. So now you see in the the communities, they are filled with aflatoxin is bad. No, the peanut is bad because they have aflatoxin. Then they have allergens. Some people get died of it, peanuts, all these kind of things that scare the community for that. I think still they're lacking of the good propaganda to really to what's the benefit peanut, what's the really good nutritional value for that. We need to get a positive way to really outreach to the community, to the marketing, to the system. Yeah, that's what I think we need to do something about that.
Great, thanks for adding that, JS. Our fourth group was led by Jeremy, and I think Danielle Fonseca is the rapporteur for that. They were discussing forging partnerships between USAID in-country programs and innovation labs, which I know is something that we all need to do more of. So Danielle and Jeremy.
Yep, thanks, Jessica. I'm gonna start the reports, and Danielle will jump in if I miss anything. But I think, the big takeaway that we had from our discussion was that this integration and collaboration really needs to be put in place very early on, not just in the design of a project working together, but in the designs of a research project being funded by an innovation lab or funded by USAID, as well as the development project itself. That really needs to be a chosen thing to be happening in both projects on both sides. And that was summarized in the AgDiv project design as well. And I think that's why we're here at this step. Somebody said AgDiv will need to work with innovation labs. And because I think that needs to be in place, because there's a few things that are missing at this point that potentially lead to this disconnect between research projects and development projects. One is the funding mechanisms that research projects or innovation labs have been calling for research projects, not development projects, just like missions are calling for development projects, not research projects. And so it really does need to be a meaningful change that happens at that level. And then kind of another very simple one is, and easily understood from both sides, it's very hard for researchers to know what development projects are doing in a country, and it's very hard for development projects to know what researchers are doing here. So there also needs to be a concerted effort to open that line of communication in an active way and in a continuing way. Another good point that came up, kind of at the individual level, is making sure that research questions and development questions align and are given the same value. Thinking, we talked about this a lot yesterday, but thinking about what the research outputs needs to be, making sure that that publishing output is still an option for researchers, but that development outcome of an activity that they could take to a group of farmers or take to a market is there as well. and the importance of both sides investing in people that are interested in playing in that role. You know, I think that's really from a mission standpoint as well. They need to have somebody who's aware of the research community and how that operates to, again, kind of serve as that translator role. But really it needs to be a meaningful and a concerted effort to make this change happen. Sorry, and I think
dogs like to participate.
Yeah, exactly. And you know, I think we are in a good position to be discussing this because we are at this point that USAID is looking to make this change. And it might seem daunting at the moment, but having this communication and putting the resources to it, I think will go quite far. And so if there's anybody else from the group who would like to contribute as well, this would be a good point. If I missed anything, I'm sure I did. We had a good conversation. So.
Yes, it's okay, Joey. Very nice report, and probably just add something is that we do have some opportunity to meet people from the mission, but it's very rare opportunity. For example, could be at the Peanut Innovation Lab annual meeting in one country, but it's just one time, because if you had the annual meeting in Senegal, the next annual meeting will be in Uganda. So you will meet people from Uganda, but for me in Senegal is not as relevant as meeting people from Senegal. So and at this meeting we are also focusing on presenting what we are doing, but not on hearing what the mission is doing and what they want us to do with them in the area that where they are working. So we need more also mechanisms for meeting, for exchanging, and for knowing what each other is doing to better program what we want to do together. And this is also one of the points Jeremy was mentioning in his report. So thanks.
Yeah, I think, Danielle, I think that's a critical point, and something that the ME will think about. I think how we can sort of get a better sense of what the missions are up to, and get you involved in those discussions as well. And it does make sense also that you know, so much of this work is country specific, and so finding ways to get everyone who's working in a country together to communicate is important.
Yeah,
the next group was oh, sorry, Jeremy, please go ahead.
No, just saying, yeah, I think that's a good summary and a good way to think about it.
Okay, good. The next group is the group that I was involved in, which I'll let Brad report on our conversation. As I mentioned before, we didn't get through nearly all the questions that we had, but we had a great discussion nonetheless.
Yeah, we had 4 questions, and the first one was, is modernization, mechanization the key to youth retention or a myth? I won't read the other 3 because we did spend most of the time on that one. I think that became our kind of main question that we talked about. So we saw, I mean, the general perception among youth is that, particularly sub-Saharan Africa, that youth advancement often means leaving agriculture, the rural areas of the rural sector. Then we talked that there are many opportunities within agriculture and within the broader value chains of agriculture, including restaurants and these types of things as you move up the value chain. And so agriculture, you know, within agriculture, there can be lucrative businesses. And so part of we talked about this idea that there's a need to change the way that agriculture is presented, so that youth can become more identified with agricultural careers. And we talked some about the use of social media for that, which I don't know a lot about myself, and that plugged in, but talked about Twitter and these type of things, and how to get more engagement of youth and more image of kind of the opportunities of agriculture within social media. We also then talked about this question of can modernization then engage and retain youth in agriculture? Mechanization was certainly pointed out as an area that can relieve the perceived drudgery and labor drudgery associated with agriculture in many places. Irrigation has the potential to enhance productivity, particularly in rainfall poor areas. In both these cases, though, there's brought up this issue, there's also the potential to enhance inequality, right? And particularly youth access to agricultural opportunities. So there's a need to tread carefully in kind of looking at those opportunities. And mechanization can also help retain youth and change the perception of agriculture as it brings higher returns. But you need access to capital, right, for mechanization. And youth often lack access to capital. They also lack access to land, which can constrain adoption of mechanization. And again, another constraint to that is the need for secure land tenure arrangements to make investments in mechanization. And that brought us up to a bit of a discussion in many areas is fragmentation of land and lack of land tenure. Both of those can be major constraints, moving in the opposite direction, moving to extensive kind of mechanized agriculture. We talked a little bit then, our last question was comparison of youth involvement in the US and sub-Saharan Africa. We did talk a little bit about there's somewhat of a difference in that most youth in the United States, it was brought up, are not involved in agriculture. And someone said food, they think food grows in supermarkets. But so there's a need to bring agriculture in the US into schools at the primary, secondary level. That can look a little bit different in a lot of low-income or sub-Saharan African countries where youth are involved in agriculture. Most youth are involved in agriculture in rural areas, but they don't really get formal training. Right? And agriculture, when they do take agricultural courses in school, it's often, someone pointed out, particularly in urban areas, seen as less preferred courses. So there's a need to, both in the US and sub-Saharan Africa, to think about revision and revitalization, I think is more the word of agricultural curriculums, particularly primary, secondary levels was brought up. And then we just kept coming back to this issue of the need to change the image of agriculture. So youth are really searching for productive and meaningful livelihoods, right? So within that search to quote, make farming cool again is kind of, how do you bring that in and make it a kind of an attractive livelihood strategy for young people? Anyone else in the group, please feel free to, I tried to synthesize as we were going along, but add it.
Yeah, I think that was a great synthesis. And I also took some notes, and I know Brad did, that we'll plan to share with everybody. And I would also encourage all of you in, when you have opportunities to go out into the field or to interact with young people, really of any age, down from primary school all the way up to college students, to have them shadow you work, to come, spend a day in the field with you, to go and spend maybe an afternoon giving a presentation at a school. I think the more that we can all do collectively to show off our own excitement about, about agriculture and the diverse opportunities that are available, and its critical place in all of our everyday lives. I definitely encourage you to do that. We've got 2 more groups to report back, so I'll let us do that. I think, at the next Rapporteurs page, Helga talking about gender responsiveness, adding gender responsiveness to research, some of the successes and failures that we've experienced.
Yeah, thank you. We had a very lively discussion in our group, guided by the 3 questions that we tackled on and off. What's your experience in including gender responsiveness in your research? What needs to be done to better include gender considerations in research? And groundnut is often called a women's crop. What are the implications for research? I'll let Paige summarize and encourage the group members to add anything that they might want to add. Thank you.
Thank you for some great questions. And I think hopefully we had some rich discussion. Hopefully I can do it some justice, but hopefully my group members can look out for things I missed. So when we're talking about the experiences and findings of the group, some kind of great examples of incorporating gender responsiveness into research, thinking about gender differences in access to resources. We talked a bit about land ownership, and not just the fact that women have less access to land, but also when they do have access to land, it's often farther away and less viable. We talked a bit about, there's some work being done in the group looking at DHS data, analyzing differences in birth certificates and how that credential is a barrier for women to achieve land tenureship. We also talked about time poverty, that, you know, the project that I'm involved in, and the gender differences in time constraints. And then thinking about differences in division of labor and how typically women's responsibilities are heavy in the household and what we call, you know, reproductive labor, and how that might influence their ability to participate in agriculture. And then lastly, there's some work being done on weeding and women's involvement in weeding, and women and children's involvement in weeding, and how interventions that address that can alleviate that burden and actually allow for children to be able to return to school when otherwise they'd be participating in agriculture. In terms of the next question, which was how can we better include gender in our work? We talked a lot about different perceptions of women's participation in peanut production or agriculture more broadly, and you know, the differences between men and women's perception of women's participation in labor, and how oftentimes women's work is invisible. We talked a bit about having the need for showing evidence-based and data-driven research that might flip those perceptions on their head. You know, that we really need to address constraints in household labor, recognize our own biases and how they might influence marginalizations of different groups. You know, following up on the great conversation about youth, thinking about intersectional approaches and how gender and youth might intersect. And then how, you know, when women might have improved access to resources, the entire community can benefit in terms of food security or livelihoods. And then right as the break was starting, or right as we got pulled back, we had some nice discussion about women's empowerment and how bargaining power within a household can influence gender equity. And lastly, this is a tricky but an important topic when we're thinking about the implications for considering peanuts as women's crops. You know, there's this tension about whether or not we're perpetuating stereotypes, or we are kind of actually using peanuts as an access point for women's involvement in agriculture. A lot of it has to do with power and control, and those power differentials can dictate a lot in terms of who's able to participate in different crop productions and also who's able to benefit from it. And so we need to be cognizant of that when we're thinking about the commercialization or market access for peanut production, as you know, oftentimes women's crops can become co-opted or over controlled by men. Also, again, thinking about this intersection between gender and youth, you know, peanuts might be considered women's crops, but in some cases it also might be young men who are participating in these kind of secondary crops. And then, you know, discussing the differences between traditional men's crops, which are, you know, the cash crops or the starches, and how women's crops can often be seen as superfluous, but they might have huge nutrition benefits, as we've talked about, and also dietary diversity in that as well. And so then, you know, how can we really engage both men and women in our activities, and how are we making sure we are addressing all of these complex issues in our programming? So a lot, but it was great discussion, and I thank all of the group members for all their participation.
Thanks a lot. Yeah, for that great summary page. I just wanted to mention one point that was particularly interesting to me. The group, Leland's group, has not yet started their farmer field schools, but from their experience in Honduras, they were reporting that, for example, because, you know, groundnuts are considered a women's group and maybe a second priority crop, that there might be a need to include technical issues in the farmer field schools to attract men to attend, you know, and then include the gender aspects as well. So I thought that was a valid point to mention to the whole group.
Definitely, thank you. There's a couple questions and comments in the chat too, which I would encourage Paige and the others to look at, and I will as well. But I do want to be cognizant of our time, and we have one last group to report back to us, I believe. That was, yes, our discussion about this very vexing, difficult issue, the technology pipelines and PLCs, product life cycles for social science projects. That was led by Danielle, and I forget who the rapporteur is right now. Soraya.
Oh, right. So Daniel was able to join us up until about lunchtime, and then he had to go off to another obligation. So I kind of handled the small but very active discussion that we had, which, you know, really wasn't so much around pipelines and product life cycles. It's really about how do we account and attribute and capture all of the great innovations that all the social science research has done. So I think the group tried to look at, you know, what are kind of examples of that, who are the adopters of that, and then yes, is a product life cycle the appropriate vehicle to try to capture that from all the research that we're doing. And so I'll let Soraya give a summary of what we tried to discuss.
You're muted, Soraya.
Thank you. I thought you unmuted me. Okay, so it was a very interesting discussion. Thanks, Dave, for the introduction. So we started from really the beginning in terms of what examples, what was asked was what examples of social science technologies, and that threw everybody off because what's a technology in the social science? So we had to go back and actually rephrase the questions. It's actually what are the research outcomes, the indicators, because 25% of the funds of the Peanut Innovation Lab goes to social science projects. And as David says, it needs to be reported in some way, and not much has been reported. So we need to report and make a way to make it visible. So technology is not the best way to describe it. You know, the social sciences actually help us understand the social context of the realities of people we are working with and working for. It helps identify the needs, the design, the physical, the research, the biological physical research do, and ultimately help adoption of the products, the practices, the cultivars. So the social science try to capture the human behavior and change it for that better adoption once the behavior has been changed or they've been sensitized to the good of the products. And also there has been, somebody said, well, the social tools and technologies can be a bridge to the technologies and to local capacity building. So it needs to permeate all the phases of the product life cycle. So there are some examples of outcomes or tools that social sciences use is the Swabble, the communication, the little videos that really can reach people and help research the effectiveness of the extension. The photo voice technology, get the technologies to change behavior of people. And also, there was a mention that knowledge can also be applied to other product developments, not just here in the Peanut Innovation Lab, but can be used to other researchers. The second question was who benefits from the social sciences here? And we decided there's a broad range of people or of entities. So not just the beneficiaries, the local beneficiaries, the people who are going to plant and use the cultivars, whatever, but also other researchers, the government, policymakers, so there is a very broad range of recipients of the knowledge that is being created. And the social sciences can help the we call it physical scientists, develop better products, advise early on in the phase to develop an appropriate product to be developed, how to get the product out there, how to, like a marketing tool, have to think about the consumers. And the last question was, can social sciences be appropriately defined within a framework of product life cycle? I'm rephrasing the question actually, but that's the gist of it. So, the social sciences knowledge is iterative. You learn something, then it goes back into the cycle many times. So there are similarities between the social sciences knowledge and the physical, biological sciences. But I think it was felt that product life cycle may not be the best model to think about the social sciences. But we didn't get to an answer what would be the alternative. And I invite the colleagues who were there to chime in, because that's my take on all the very exciting discussions we had.
Great, thank you, Soraya. Does anybody else from that group, Dave or anyone else, want to make some remarks?
No, I think it was a very good summary, Soraya. I'll let others comment as well. The only comment that I would make is that, I think that we as an innovation lab are in a unique position to be able to try to help USAID think through this issue, which is one reason why we put it on as a breakout room, even though we know it's a very difficult one, because we actually have research projects focused on social science research. And many other innovation labs just incorporate these areas into existing research projects. And so I think that's why we have a pretty unique and opportunity to really help them think this through. It's clearly not an easy one, but I think we can really give it a good stab at it. So appreciate all the start of the dialogue. But maybe Tom or others that were in there, Cynthia, might want to comment.
Well, if not now, I know that it's a conversation, yes, as Dave said, that we will be continuing to have. I think we've pretty much reached the end of our time, and I know it's been, oh, we do have a poll, excuse me. Thank you.
We can't end our meeting without a poll. We start them with a poll, we're going to end the meeting with one.
What is the best way to reach groundnut farmers about improved technologies? In person, that was my vote too. In person, cell phones and radio look like the top candidates, which makes a lot of sense. I think if we can, in-person extension would be a great job for young people in rural Africa. So if we can find ways to invest in that, I think that would be great. And obviously also in the gender composition of those teams as well. So yes, like I said, we are reaching the end of our time. I don't really have any other closing remarks. I'll hand it over to Dave, other than just, it's always really an exciting and enjoyable, set of meetings with you guys. And I'm very happy and proud to be in community with all of you. And obviously really hope that we get to do this in person next year. So thank you.
Great. Thanks, Jessica, for handling the day sessions and for everyone for participating, particularly in what I think was a great way to end a really exciting 3 days, is to have 30 minutes or a little bit longer to really focus in on some of these key issues. As I said in our beginning remarks, this was going to be a different meeting than what we might have thought we would have as an annual research meeting. We didn't focus as much on all the great, exciting innovations that we're all producing, but we tried to bring up some of these key issues that particularly we as a management entity keep hearing about in our interactions with USAID and other partners and with US missions. And hopefully we kind of did a good job on at least making sure everybody now is more aware of them. And clearly it's not, we we began to touch some of them pretty well, but we still have a long ways to go. So hopefully it's the start of a good set of discussions and dialogues that we can have. I think we will obviously be sending out a survey here shortly. Kristen will probably take care of that over the course of probably tomorrow or so. And we encourage you all to then provide your feedback. And that's the best way we can figure out what went really well for the meeting, what can we improve, and what do we want to make sure that we keep continuing. We'll also be recording. We recorded all of our main sessions. I don't think we didn't record, of course, any of the breakout sessions. Unfortunately, those might have been the most interesting ones to have a recording of. Hopefully we did capture the proceeding or the discussion in those, and we'll make all that available. So we'll make sure that everybody has access to the recordings in case you missed a session or want to go back and listen to it again. I think we're also hopefully pulling together all of the presentations. We'll have the documentation from the breakout rooms and other things that were produced during the course of the 3 days. And so Kristen will be sending out everyone a link to all of that so that we can all benefit from it. As I said, this really hopefully was an opportunity to really begin to think about and put on the table many of the different issues that we're going to have to be discussing over the remainder of our innovation lab. And so we're looking forward to that over the course of the next year or so. But also, I think we know that we're going to have other mechanisms to communicate, particularly all the great innovations and research that we're conducting. So you will be hearing from us as we begin to organize webinars around the different themes of our research. We heard some of them over the course of the 3 days, but I think we have a lot of different themes that we could put together where we can start to tell a wider range of stakeholders what we're learning, what we're producing as outputs and products, and begin to tell the world about that. So we'll be pulling those together. It was really fortunate that we were able to have 3 of our grad students make their presentations today. I think we all enjoyed that, not only because it's great to see students present really high quality work. And to me, it was exciting to see that we have students producing quality work in Africa. I think that's a real credit to African institutions and the ability of being able to have students now trained in their own home countries with high quality work. And I just point out that I think there's something like 13 of our graduate students are actually presenting at the American Peanut Research and Education Society meeting next week in competitions for other graduate students. And so I think that's pretty exciting. There's another 3 or 4 scientists among all of our African colleagues that are actually making presentations. And I couldn't count how many posters are being presented, but I think that's a really exciting outcome of the work that we're doing to really support the future scientists and researchers and partners that we're going to need. But we do want to make sure that all students can give us updates. So we'll be looking at how we can do that through student presentations as well going forward. And of course, we're already beginning to start to arrange when we can meet with each of our projects as a management entity, and hopefully we can bring in some of our external advisors to hear more details about the work and begin to plan next year's work plans and begin to think through what our product life cycles that we need to be considering as we go forward. So a lot happening, and it's going to be hopefully a very exciting next couple of years in the program. So to me, I think we accomplished what I had in mind for an annual meeting. I think the management team spent a lot of time thinking about what was the right format. I think we're pretty pleased, but we still want to hear from everybody as well as to your thoughts, etc. But I think it was a very successful 3 days. So really the only other thing I need to do is thank everybody Definitely want to thank all the participants. I mean, I was surprised, I wasn't surprised. I was pleased today to see that we even had almost over 90 participants on the third day. Each day we had about 115 registered, and some days we had over 100 of those participating, which is really exciting to see. It just shows how much people are hopefully, committed to the program and interested to see what we're all doing collectively together. So thanks mainly to all of you. Thanks to all the speakers. I think they all were excellent. We really appreciate the time it takes to put together a presentation, and particularly our 3 invited speakers. I think that's sometimes the most exciting. You get a different perspective, you get some different ideas coming on the table, which kind of stimulates our thinking going forward. Thanks to the moderators and the rapporteurs today. I think that was probably one of the highlights of the meeting for me, was to be able to have small group discussions. We knew it probably would be, and we could have probably run the entire meeting as breakout rooms, but I think it was also exciting to see how much you can discuss and accomplish in 30 minutes. A little bit of focus time gets you focused on a topic. So hopefully that's something that we can keep thinking about how to do that in the future. And finally, I just want to thank all of my staff. They put in a lot of hard work. Allison put in all those exciting videos and welcome videos, and that's the credit to everybody who submitted them. And of course, a big thanks to Kristen, who dealt with all the logistics. It's been a lifetime to learn how to run a meeting in person, and we've only had like a year to learn how to do it by Zoom. And I think we're all still learning how you make a Zoom meeting as interesting and exciting and as productive as we can do in person. But you did a fantastic job, Kristen, behind the scenes, making things flow well. And so we really appreciate that.
Well, thank you. And I have a wonderful team and wonderful colleagues across the entire world. So help make it happen.
So anyway, thanks a lot. It was great. We'll hope that as I said, we hope now that we're going to be able to start seeing more people in person. And definitely we're looking forward to the meeting next year. We're not quite sure exactly where it will be. We'll start to think about that. But we're hoping that at least a good number of us can get together in person, and we can have a really good discussion together. I think we'll learn that we will also be able to hopefully offer whatever meeting we have in person also virtually, so we can have a strong participation across the whole program. But I think we're all looking forward to that time to at least see someone in person and have those types of interactions as well. So thanks a lot again, and we'll be in touch, and we hope to see everybody at our next annual research meeting.